Biology Insights webinar, titled Assessing CAR T Cell Therapy in Vitro, Glioblastoma Organoids. I'm Lauren Coyle, an editor at BioInsights, and joining me today is Meghan Logun, who will discuss promising CAR Ts, dual targeting CAR T cells for recurrent glioblastoma, using patient-derived organoids to model treatment response and correlate with clinical outcomes. After the presentation, we'll have a live Q&A session, and we invite our audience to pose their questions to our speaker using the Ask a Question box at the bottom of, and we'll try to get to it during the session. Now, I'd like to introduce our presenter. Meghan Logun holds advanced degrees in animal science and neuroscience with research experience in glioblastoma and cellular immunotherapies, and currently serves as a postdoc and regulatory oncology product reviewer at the NIH FDA, specializing in cell and gene therapies. So without further ado, I'll hand over to Meghan to kick us off with her presentation.
Thank you so much for that introduction. Today I'm going to be talking about one of my postdoctoral projects that I participated in at the University of Pennsylvania, where in tandem with our ongoing clinical trial for a bicistronic CAR T cell, I was able to use patient-derived glioblastoma organoids to look at CAR T cell therapy in vitro. So for those of you who are familiar with glioblastoma, you know how devastating a disease it can be and how it remains a massive unmet medical need. It is the most common primary malignant brain tumor in adults. Patients have almost 100% chance of recurrence, and even with all of the standard of care therapies thrown at it, the average patient survives less than 1 year. And you might have seen a lot of recent press about the advances made in cellular immunotherapy, with one of the biggest ones being chimeric antigen receptor T cell therapy, whereby they take the patient's own T cells and transduce them to express a T cell receptor that then targets a tumor-specific or tumor-associated antigen. And even though these have shown really great success in hematologic malignancies, they have not seen the same success in solid tumors, and more specifically glioblastoma, with one of the biggest issues being that glioblastoma is prone to antigen escape and is a very heterogeneous tumor. And so we decided as a CAR T cell lab who targets glioblastoma to make a bivalent CAR T cell. So that means that our CAR T cells have two different chimeric antigen receptors expressed on the surface, one targeting an EGFR variant 3 tumor-specific mutation, as well as the commonly overexpressed IL13Rα2. And so separately, these two antigens are prevalent across 50 to 60% of primary GBMs. And so this is the product that we are currently testing in a first-in-human phase 1 for recurrent GBM. So we have taken these bivalent CAR T cells, and we have patients who have already received diagnoses of recurrent GBM. They receive a surgical resection here at Penn, and then post-resection, they receive CAR T cell treatment in one of the three different dose escalation in one of the three doses, and then are followed for long-term survival and bioactivity of these CAR T cells. And so one of the things that interested me as a researcher is that we have these patients coming in receiving surgeries at Penn, and then that tissue is being used to turn into patient-derived organoids. And can we use those in combination with other resources generated from the trial to see if we can create any biomarkers or help with interpretation of how these patients are actually responding in real time to that treatment? So one of our collaborators, the Song Lab at University of Pennsylvania, they worked with us previously to publish a paper on what we call our patient-derived glioblastoma organoids, or as I'll call them, GBOs, moving forward. And so this is a pipeline by which we're able to take resected tumor tissue from GBM patients and culture them long-term as these multicellular organoids that retain a lot of the original tumor architecture. So the GBOs are obtained fresh, microdissected, and then we are able to culture them out over, and we have data up to 12 weeks where they're cultured before or being stored and biobanked for long-term storage and potentially future studies. One of the really cool things about these GBOs is that in addition to maintaining a lot of the cellular heterogeneity of GBM, of the parent GBM that is, they also retain some of the tumor-associated vasculature and the hypoxic zones that are hallmark to GBM, which is something that's very hard to reestablish from in vitro cell cultures, for example. And ultimately, these GBOs are really invaluable because in addition to just maintaining the diverse heterogeneity of different cell types from the parental tumor, we can actually map over transcriptomics that from the parental tumor through a 2-week culture of becoming the GBOs, that a lot of the original cell clusters are maintained as seen here in the UMAPs, and that these correlate very strongly to the original tumor signifiers that from which they came. And so what we wanted to do was see if we could took those GBOs from our patients in this trial, and if we took some of the autologous patient CAR T cell product that we are testing as well, can we perform in vitro assays that would give us any more information on how the patients are responding in real time? And so, as you can see with the CAR T trial, these patients are getting a multitude of assessments performed, such as radiographics. We're drawing CSF from these patients in order to look at CAR expansion, as well as looking at cytokines present in the CSF as well. Now, on the GBO side, after they've received surgery and we were able to generate their GBOs, as well as the patient product is manufactured, we were able to take those two things and co-culture them and then look at parameters such as tumor cytolysis, CAR T cell activation and engraftment, as well as cytokine expression in the media of these assays to kind of look and see if we can deduce or if they even look similar to what's happening in the patient. And so, again, these assays that are going to be described here are going to include the GBO and autologous CAR T cell product impedance assays, cytokine ELISAs from these co-culture impedance assays, as well as immunohistochemistry of co-culture GBOs retrieved from the impedance assays, as well as flow cytometry of the GBOs and the CAR T cell product retrieved from these assays. And why this was really important for us to do is that it could be done in real time alongside the patient treatment. So what that actually looks like in terms of a physical timeline is that the surgical section takes place, and then CAR T cell treatment is begun within about a month of that surgical resection. And at day 0, at time of treatment, we have already also produced the GBOs and received the clinical excess clinical product so that we could be starting their co-cultures alongside patient treatment timeline. And since these GBO co-cultures that are performed, they run about 6 days long, and our first patient MRI isn't even until day 28. So we're actually seeing evidence of cytolysis by the patient product, within a fraction of the time it takes for us to see our first patient MRI. And so the real workhorse of these assays is the impedance platform, at which we partnered with Axion Biosystems, and we used a Maestro ZHT impedance system to look at. And I cite their application note here, which is what gave me the original idea to perform these real-time assays in comparison to a chromium release or something else that was more of an endpoint assay, because we wanted the real-time output, and they had designed protocols whereby you could actually do this on spheroids in their side-of-view 96-well plates as opposed to 2D mono layer cultures. And so what we can see is that they show that you can have different densities of spheroid added to their plates. They create a impedance or a resistance signature that we can then add effector T cells to, which affects that resistance and can be converted into a cytolysis output. And so what I did was, based on this application note, I took GBOs, I plated them into PDL and laminin-coated 96-well side-of-view plates, and allowed them to cure for about 24 hours before adding the effector CAR T cells. And I separated these into 1 GBO per well. They're rather large, and they come out to be around 100 to 200,000 cells per organoid at time of placement. And so these are pretty large and cover a decent bit of the surface of the side of view plates and create a nice, robust resistance signature. And then we add the autologous CAR T cells at a 1:10 effector to target ratio. And these co-cultures were monitored for 6 days before cells were retrieved. This includes over time at relevant time points. I did pull out media from these assays to do cytokine ELISAs. And so first looking at what these GBOs looked like, and I'm showing you here data for 6 patients from the trial. We show that all 6 patients, we were able to isolate the GBOs, and that when we look at the actual IF of the patient GBOs, before even starting, we had to validate that the target antigens were there and present in the GBOs, and this was maintained from the parental tumor. Now, as a side note, in the trial, clinical criteria for enrollment only included the amplification of EGFR, and we did not have to analyze the IL13Rα2 signature. This was also kind of a sanity check for us to make sure that both antigens were present in these GBOs and in the parental tumor. So for each patient on the left, you have IHC showing DAPI, EGFR, IL13, and a merge of the post-resection tissue before processing into GBO, and then after 21 days of processing into GBOs. And all you need to take away from this slide is that we have nice, robust presence of both target antigens in every single GBO. And so thereby we know that when we introduce these into our co-cultures, that there should be target for these CAR T cells to find. So as we move into the data from the impedance co-cultures, what we can see is that from our first 6 patients, if you take their GBOs and co-culture them with their matched autologous patient product, they all have differing levels of cytolysis over time, and that this is significantly different from GBOs that were untreated in all 6 patients. So there was levels of cell killing present, but across each patient, there was obviously different amounts of cytolysis in terms of a different response, which was to be expected in terms of patient-to-patient variability. And so this becomes more important as we move into what's actually happening in vivo. So if we look at the same 6 patients, the data of the CAR expansion, and this was published earlier this year in our group's Nature Medicine paper. Again, on the same first 6 patients, we probed the CSF to look at the amount of CAR copies per microgram of genomic DNA present to ensure that the CAR T cells were actually expanding in the CSF and that they were actually present. And what we saw is that there was a huge spike within the first day or two after infusion, leading to this maintenance or slight drop off of the amount of CAR copies found in the CSF. Now, this adds a lot more color to why we see some differing levels of cytolysis. And one of the really interesting things found from this study is that when we look at the percent cytolysis observed in the first 24 hours in those impedance assays, it strongly correlates with the peak engraftment data from these same 6 patients. So this could be an early indicator of, even though we saw what seemed to be very variable responses in the impedance data, this actually correlates very strongly with the amount of CAR expansion in that short term. So it gives us a little bit of an insight into how strong that CAR expansion was across those patients. And to ensure that what we were seeing is on-target killing, we orthogonally confirmed this by we took GBOs out of those impedance assays and did cleaved caspase 3 immunostaining after the 6 days of co-culture. And so what you're seeing here is that between untreated and treated for each patient 1 through 6, that we saw this massive expansion of CD3 T cells, as well as presence of cleaved caspase 3, which is an apoptotic marker. So this is showing that these CD3, the T cells that we are incubating with these GBOs, are not only invading into the GBO, but also causing cellular apoptosis as a result. And so this was confirmed to be significant across all of our patients at endpoint. And so to confirm that these cytotoxic events were actually due to on-target antigen recognition and not off-target toxicity or maybe native TCR recognition by the patient product, we performed a couple control experiments using an off-trial patient GBO, where this patient who received their surgical resection and then unfortunately had to be removed from the trial for other medical reasons, We received their GBOs and their patient product, and we were able to do impedance assays where we looked at them over the 6 days with untreated, their autologous matched product, a CD19 off-target CAR made from their the autologous T cells, as well as the untransduced autologous T cells. And what we can see at endpoint is the significant difference between the autologous product that contained the on-target CAR versus off-target CD19 or untransduced. And this was also reflected in the cleaved caspase 3 confirmation, the fact that we also saw no Ki67 content in the CD3 cells that were found in GBOs that received untransduced T cell treatment, meaning that none of the T cells that were found in those GBOs were expanding as a result of activation. And then I think possibly the most impactful piece of data from this control experiment showing that in GBOs treated with on-target CAR, off-target CAR, untransduced T cells, or even in the T cell only cultures, none of them created a cytokine expression profile that included interferon gamma, IL-2, and TNF alpha, unless it was the on-target CAR with GBO. So the off-target CARs were not contributing to any cell killing, and they're therefore releasing any cytokines. So to further elaborate on the on-target activity, we also performed IF to look at the amount of the actual antigen present in the GBOs before and after treatment. So what you're seeing here is that for each patient, we have untreated and treated GBOs with presence of the EGFR or IL-13 receptor alpha-2 antigen shown respectively. And what we see across almost all of the patients is that there are significant reductions in the amount of EGFR as well as IL-13 receptor alpha-2 post 6 days of treatment. And in addition to IF, we also wanted to look at the double positive and double negative populations. This is really impactful because when we pull the GBOs back out from these co-cultures at 6 days, there's still a large amount of cells there. And these are the cells that we would assume in vivo are leading to recurrence because they're either avoiding detection, they are avoiding destruction, or they are double negative from either of the antigens present on our CAR T cell. And so what we did was, in order to look at a more complex populations, we took the GBOs out from the impedance assays after 6 days and performed flow cytometry so we could really look at those double and single positive and negative populations. And what we see for each patient is that, and I'm going to look at the summary data here, is that for the double positive populations after treatment, there is a significant reduction in all of the double positive cells, which is what we would expect, as well as a significant increase in the double negative population. So these are the cells that would be theoretically escaping treatment, as well as a large amount that are driving EGFR positive, single positive populations down. But interestingly, we did see an increase in the IL-13 receptor alpha-2 single positive populations, which in conjunction with other data that we're pulling from both patient data and other data not shown here, but in the paper that you can find on bioRxiv, we think a lot of the CAR activity is being driven more by the EGFR than by the IL-13 receptor alpha-2. But again, this is all still being studied and will continue to be studied on what the changes in these single and double positive populations mean as we increase our patient numbers and in the amount of GBO assays we're doing. So lastly, one of the coolest, most impactful pieces of data that we got from the patient impedance co-cultures is that when you take out media from these co-cultures, they exhibited commensurate trends in cytokines as seen in ELISAs after CAR T cell treatment that mirrors what we see in patients. And so what you're seeing in panel A is that for each patient in each cytokine, the GBO spike and then plateau or decrease in the amount of cytokine present directly mirrors what was seen in the patient CSF values. And this is for almost all 6 patients And the fact that not only are the trends mirroring, but the actual concentration of cytokines that were in the wells compared to in the CSF being commensurate was pretty incredible to see. And especially when you look at panel B, when we look at these three key cytokines over time in the GBOs versus the co-culture, a lot of these were trending very similarly. And lastly, there's not many are significantly different from each other over these first 6 days. And so these cytokine, the cytokine release that's happening in the plate is very similar to the cytokine release that's happening in the patient CSF after treatment. And so this could be a good additional real-time assessment to give you more color into what's happening in the patient. And just to look at this in another way, what we see here is the data shown in our GBO study here compared to the data published in the Nature Medicine paper from the patients, where they look at, again, interferon gamma, IL-2, TNF alpha, and in the inclusion of IL-6, which we hope to include in future works, but that these the amount of cytokines and the peaks present all change the most within the first 7 days, which seemed to be a critical window of looking at bioactivity. And then in the patient, they seem to plateau within the first month. And so ultimately, since we know we can take these GBOs out a little longer, I think that will be included in future studies to see if we can maintain that kind of high degree of fidelity between the two over a longer period of time to get more information out of these GBOs, but that remains to be kind of looked at. And so a couple conclusions from this work is that GBOs that we generated from our patient resection tissue can be used with pretty high fidelity to represent certain features of patient GBM. This is not perfect and it's not meant to be, but it works us one step closer towards looking at ex vivo biomarkers of what's happening in real time, and that the ex vivo cytolysis data generated here from the Maestro ZHT was very highly correlative with patient response to treatment in the early critical time points of bioactivity. We also saw that the ex vivo assays performed here in the side of view plates had very proportionate cytokine dynamics as the in vivo patient CSF at the same time points. And so this will be critical as we move forward and are looking at more and more patients and hopefully looking at longer time points to see if we can learn even more information from these ex vivo assays that will help us understand, recurrence dynamics or responder dynamics in these patients. And lastly, some immediate future works that we plan on doing to expand this is, of course, expanding our patient cohort just to get more data, as well as looking at dose ranges. One of the more interesting things that we didn't look at is that we didn't change our effector to target ratios throughout these assays. And so one of the things in the future would be changing up effector to target ratios in terms of, can we mirror patient dosing dynamics in these Axion killing assays. The other immediate future work is that, like I mentioned, when we looked at antigen presence before and after treatment, is that those populations that are left over after 6 days of treatment, especially because those are the cells that are left over after that critical first week of bioactivity of the CAR in the patient, how are those contributing to recurrence? And can we use rechallenge assays in the Axion impedance platforms to look at how those resistant populations survive and maybe how we can start trying to target those better. And with that, I'd really love to thank my lab, everyone who contributed to this work, made it so much fun, and our collaborators in the Song Lab who were critical in harvesting and maintaining GBOs, as well as data collection and analysis. Anyone who worked on the clinical side with the clinical trial, including Steve Bagley, who is the PI for this ongoing phase 1 at Penn. And special thanks to Axion Biosystems for their collaboration in helping with instrumentation and experimental design, as well as our funding sources. And thank you, instrumentation, for having me here today to talk about this work. And I would love to take any questions you might have.
Thank you, Megan, for that absolutely fantastic presentation. So as we said, we will now be starting our Q&A. So please do keep your questions coming in. And Megan, if you'd like to join me on camera. Wonderful, thank you. So yeah, we'll just kick off with our first question here then. How long can these organoids be maintained ex vivo until they no longer represent patient tissue? Oh, I think you might be on mute there, sorry.
I was, thank you. I was saying that's a great question, especially because I know that a lot of other people studying cancer tissues have kind of the same problem. And right now, I would say that we have data showing that key critical aspects are maintained through 12 weeks of active culture. And then also GBOs that have been biobanked before that point, frozen down and thawed well beyond a year, have been thawed and have been shown to also keep a lot of those same key things that we look at. But as we kind of look to expand that longer, I know that we're going to be probably looking at 24, like 3-month long data points at some point.
Wonderful, thank you so much. And our next question here is, did you dissociate the GBO into a smaller size for impedance assay?
So for the impedance assay, no, they were not dissociated. What we found was that the GBOs were large enough to actually, when you put them on those coated plates, to form adhesions across the bottom of the GBO. And so they had an impedance signature that was large enough to then look at changes when you added the effectors. We did, however, dissociate them specifically for flow cytometry, since that is more of a single cell based analysis.
Amazing, that was great, thank you. And you mentioned the hypoxic environment of the TME. Do you ever look at the T cell phenotype of your CAR T cells before and after being co-cultured with GBO? And what impact do you think the hypoxia had on the efficacy in your model?
Yeah, that's a great question. So we do have the data for the T cell phenotyping of the CAR patient product for all the patients. I didn't go into it here since it's limited time, but I definitely recommend, if you're interested in that, check out the Nature Medicine paper, as well as this paper, which we actually just received word that it was accepted. And so it should be hopefully out as well soon. But as far as what impact I think that it had, I think in future studies, I don't have a good answer for this right now based on what we've seen, because we didn't specifically look at hypoxia, but I know in the future we will be more deeply looking at, of those CD3 positive T cells that penetrated into the organoid, the depth of penetration and that effect on the T cell phenotype and persistence will be looked at, as well as possibly putting the GBOs in hypoxic chambers and doing co-cultures actually within the hypoxia chambers as well, to kind of even more bring the microenvironment into play.
Amazing, thank you. And can you elaborate on some of the differences in using this platform between 2D and 3D?
Yeah, I think that, that's a great area of comparison. And I think that here, what we really wanted to do is that we know, and we have a lot of data on our CAR killing in the 2D adherent substrate models. And what we really wanted to do here was look at the difference that that the 3D microenvironment made in terms of if we could measure killing in a bulk environment where the T cells have to really work for it, compared to just laying them over top of the 2D platform, since the 3D is more realistic to the in vivo.
Wonderful, thank you. And does CAR T cells exert killing effect only when two targets, so the IL13Rα2 and the EGFR, are binded?
Yeah, so that's a great question, and the short answer is almost always. We have data in previous papers showing that off-target toxicity for these two targets specifically are low. If you remember, I said the EGFR, we're not binding wild type, we're binding a specific tumor-associated mutation. And then with IL-13 receptor alpha 2, again, it's mostly prevalent in that tissue in the brain. So we see very little off-target toxicity in our assays that we do in other platforms.
Wonderful. And we have time for probably just one more here. So in the future, if a patient's GBOs did not respond to CAR T therapy in vitro, what could a clinician do with this information? Could they plan to add additional treatments or perform further function or genetic testing on restraint cells to find therapeutic targets?
Yeah, that's a great question. I think it goes straight to the heart of the ultimate vision for these as both predictive medicine as well as establishing some of those mechanistic whys of maybe why we see responders, why we don't. And so ideally what we'd really love is that they could be used to, like you said, give a clinician a little bit more color on what's going on so they can determine, okay, is this patient eligible or is it a prime candidate for redosing? Is it a prime candidate for maybe increasing the dose or not? Should we discontinue and try a different trial as a better use of this patient's time, as well as looking at those resistant populations to maybe figure out why this patient wasn't responding to treatment.
Fantastic. Well, Megan, thank you so much for answering all of those questions, but that is all that we have time for today. So any questions that we didn't get to, we will reply to by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. And we'd also like to invite you, the audience, to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources on the right-hand side of the screen or in the on-demand email tomorrow. So all that's left is to thank Megan once more for a great presentation, and thank you to you for listening. We hope you'll join us again soon.